This is about the inter-individual variability of testosterone doses and needs.
So welcome back to the TRT and Hormone Optimization YouTube channel. And back with us, we have Ryan Root. Welcome back, Ryan.
Hey, how are you, Dr. Devos? Thank you.
You're so welcome. So recently we had some comments like this in the video section. Why is my body not absorbing my TRT? Some people say I have to take more than 300 milligrams a week to get my levels up to a good range. So I guess we are going to talk about low androgen sensitivity or testosterone resistance. I don't know if that is exactly the same thing, but I'll let you do the talking, Ryan.
Okay, thank you very much. And again, thanks for what you do here with this channel. You have a lot of very informative interviews with people, and it really helps people a lot. And I've learned a lot, and I know others have too. So again, thank you.
You're welcome.
So what I want to start to say is that yes, the factors that determine every individual's how they are affected by testosterone is complicated. There are likely 1,000 different factors that we don't even know about yet as humans. So there are so many complexities, and because we don't understand all these complexities, the numbers don't mean much. So what I'm going to do is go into some of the, again, there's probably likely 1,000 different things that we don't understand, but I'm going to go into some of the factors that can cause inter-individual variability of TRT dosage and protocols. So the first thing is something as simple as the type of blood test can lead to variability in numbers, right? So the ELCIA blood test, electrochemoluminescent immunoassay, is notoriously inaccurate because it cross-detects other similar compounds. So notoriously, if you get an ECLIA estradiol test, it will also cross-detect estriol and estrone and give you a inflated number, right? So that's why people usually opt to go for the sensitive estradiol test through LCMS, liquid chromatography mass spectrometry, which is the kind of the gold standard, right? So if we're ordering an ECLI, or if your blood test is done just through ECLIA and you didn't know that it could cross-detect possibly DHT or DHEA, right, and give you an inflated number. So that's why some people might see an inflated number, and something as simple as the type of blood test. So the next thing is, I made some notes over here, so I'm just going to check on those once in a while. The low SHBG in TRT patients causes elevated free testosterone that can be used quickly, which can yield a low total testosterone reading. So if we have low SHBG, we have high free testosterone. Now this free testosterone can be used or eliminated relatively quickly through two ways. One is glucuronidation, which we'll go over later, and it can also quickly bind to the androgen receptor. So our blood tests are a snapshot in time, right? And so we don't get to see some of the peaks and valleys if our free testosterone was used really quickly and then dropped. We don't get to see that in one blood test snapshot over time. So depending on when the test was taken, if our free testosterone was utilized or glucuronidated and filtered out by our kidneys relatively quickly, then we won't. Then it can give us a lower reading than expected. Right. So that sometimes people will say, I'm taking all this testosterone, I have a low total testosterone. What happened? And that's because your SHBG is low, your free testosterone is high, and it was utilized really quickly. That's one possibility. Okay, so now the medical community in our current paradigm, we only pay attention to the amount of testosterone in our blood to determine low testosterone or hypogonadism, right? But we're missing the other side of the equation, which is the binding to the androgen receptors. Testosterone in the blood means nothing until it binds to the androgen receptor. So we're going to look at some androgen receptor variability, some things that can cause disparity in the testosterone actually binding to the androgen receptor. So one of these things is CAG repeats. Our DNA is made up of 3 nucleotide sequences called codons, right? And at the end of the androgen receptor, of exon 1 of the androgen receptor, cytosine, adenine, and guanine, CAG, are repeated on average 8 to 35 times. Now, there is the number of androgen receptor, the CAG repeats is inversely proportional to the amount of androgen receptors throughout your body. So if you have a high number of repeats, then you have a low number of androgen receptors throughout your body, and vice versa, right? So people with a high number of repeats and a low number of androgen receptors throughout their body will have a lower sensitivity to testosterone. So testosterone will not affect them the same as somebody with a high number of repeats. The other thing is the binding affinity of testosterone to the androgen receptor. So the androgen receptor forms and folds into this conformation that has binding points that perfectly align with binding points on a testosterone molecule, right? So now everybody's different. So there's variability in each individual's, the way the androgen receptor forms. So some people can only have a few binding points right here, right? So when you have a testosterone molecule floating through the cytosol of a cell, this testosterone, if you have few binding points, it won't be as attracted to the androgen receptor. So it could just float by, not bind to the androgen receptor, and not have its cascade of effects, right? If you have higher amount of binding points, then the testosterone is more attracted, and you have more frequently it will bind to this testosterone molecule. So the binding affinity of your androgen receptor to the testosterone molecule is important. Higher binding affinity means you'll have more susceptible, or you'll be more, testosterone will have more effect for you, right? Mm-hmm. And lower binding points, you will be less sensitive to testosterone. So these are all genetic factors that can cause a person, with 1,000 nanogram per deciliter is not as affected as somebody who has a higher sensitivity to these androgens. And everybody's different. So the next thing is dissociation of testosterone from the androgen receptor too quickly. Okay, so what happens is testosterone binds to the androgen receptor. It translocates to the nucleus where it binds to the DNA, to an androgen responsive element on the DNA. It then transcribes into mRNA, and then translates into a peptide or protein, which is cell specific, depending on what cell we're dealing with, that has a certain cascade of effects, right? So when testosterone binds to this androgen receptor and it goes to translocate to the nucleus, if testosterone dissociates too quickly, then it can interrupt this process, right? So it could either make this so the testosterone bound androgen receptor binds inefficiently to the DNA, which can cause some level of impotency in the cascade of effects, or it can if it dissociates far too quickly, then the testosterone bound androgen receptor can't bind to the DNA at all. In fact, there's one type of complete androgen insensitivity syndrome, which is a disease state where testosterone binds to the androgen receptor, but it dissociates too quickly and can never bind to the DNA to have its cascade of effects. In this case, somebody who, because you're born with this condition, somebody could be born an XY male, but because they have complete androgen insensitivity, they develop breast tissue, a vagina, and working ovaries, right? So a notorious popular example of this is actress Sigourney Weaver. She's notoriously a XY female, right? So she was born a male, but has complete androgen insensitivity and developed all the female characteristics, right? Yeah. So there's everything in between there. So there's just variability in the way for each person, the way if testosterone dissociates too quickly. So another thing is testosterone, again, binds to the androgen receptor. When it translocates to the nucleus to bind to the DNA, it recruits these co-activators that help bring it to the nucleus and help it bind to the DNA. Now, inefficient binding of these co-activators can destabilize the process, right, of this. Now, destabilizing the process can do one of two things. Either it could, again, bind to the DNA inefficiently and cause an effect on the potency of the testosterone's cascade of effects, or it can cause it can take a long time to translocate to the nucleus, right? if we have a stable coactivators, then it translocates the nucleus quickly, and the cascade of effect happens very quickly, and then the androgen receptor can regenerate quickly. If this process is very slow, then it takes time for the androgen receptor to regenerate, which means we have a lower frequency of androgen receptors, right? So inefficient process causes, for two different possible ways, it causes less sensitivity to testosterone, right? So all kinds of these things can go wrong. So here's another process. We evolved a process to defend our bodies against too high of hormones, right? So we go through this process called glucuronidation, where our body actually conjugates a glucuronic acid molecule to the testosterone or to any hormone. But we'll specify this with testosterone. This happens with all hormones. What this does is it deactivates the testosterone so that it can be filtered out by the kidneys. When we glucuronidate, it makes the testosterone, normally a hydrophobic oil soluble molecule, it turns it into something hydrophilic, which is now water soluble, and water soluble things are filtered out by the kidneys, and hydrophobic or oil soluble compounds are filtered out with the help of bile through the liver, right? So so our body is constantly doing this. Now one of the interesting things, if you have a high amount, an adequate amount of SHBG, testosterone-bound SHBG cannot be glucuronidated. So it kind of like it kind of isolates it from that, and then your cells can use the testosterone-bound SHBG, right? So that you're getting your testosterone with it, and it doesn't have a chance to be glucuronidated. High free testosterone, your body will actually increase the incidence of glucuronidation to protect, again, against the too high of free hormones. So what can happen is your body has all these processes to protect the too high hormones. If you have low SHBG, too high free testosterone, you'll increase the amount of glucuronidation, and you can actually start to have symptoms of hypogonadism again, because you're desensitizing your body to this testosterone, right? Now there is some new evidence of testosterone glucuronide being able to be deconjugated and reactivated in the gut, right? So some of it can be reactivated, but a portion of significance is just deactivated and filtered out through the kidneys. Okay, so that's it. Oh, and so there's different genetic factors or different genetic predispositions. Some people might have a higher incidence of glucuronidation. Some people might have a lower incidence, right? So that's a genetic factor that's just different for everybody. One causing low androgen sensitivity, and if you have low glucuronidation, you're going to be more sensitive to testosterone. Right? Yeah. So the next thing is these environmental toxins. Now, some of these environmental toxins can interrupt androgen biosynthesis. So it affects the HPTA, which would decrease the amount of luteinizing hormone or follicle stimulating hormone, and it can also damage the testicles, the Leydig cells in the testicles, which actually produce the testosterone, right? So this is also certainly a reason why younger and younger people are needing testosterone or have low testosterone, right? But however, this is about the inter-individual variability of testosterone. So I'm going of testosterone doses and needs. So I'm going to focus more on the environmental toxins and which ones can actually interact with the androgen receptor to interfere with the ligand-dependent transcriptional activity, which essentially means that they block androgen receptors, block testosterone from binding to the androgen receptor. So there's an entire class that does this, right? They block the androgen receptor. Some of them can actually make the androgen receptor fold differently so that it loses its binding domain, or they can actually affect those co-activator proteins so that testosterone can't make it to the DNA, or it's inefficient, right? So they can affect it all kinds of different ways, these environmental toxins. So let me go over a few of them. Methoxychlor was an insecticide that replaced DDT when DDT was banned, right? BPA and BBP are used in the production of plastics. Incidentally, BPA is an estrogen agonist and an anti-androgen, right? So it's estrogenic and it acts as a androgen blocker. So it's kind of a double whammy right there. Vinclozolin and prasimidone are fungicides used on fruits and vegetables. Interestingly enough, vinclozolin, the chemical itself does not have an effect on androgen receptors, but its metabolites do. Right. So here's a whole new thing that we have to look at that I'm sure the government and the EPA is not looking into. Not only do we have to make sure that the individual pollutants are not causing damage to our bodies, but we also have to look at each metabolite to see what they're doing to our bodies. Because like in the case of this one, the chemical itself doesn't do anything, but the metabolite does. It becomes an anti-androgen. Linuron, which is found in herbicides to control weeds, and lindane, which is an insecticide. So here's just some of these chemicals that are in our environments. So, we've examined a lot of these environmental toxins that are anti-androgenic. So the question is, how do we avoid these? And this is part of the problem, is that they're ubiquitous in our environment, right? So you could make sweeping life changes, but they're still in the food we eat and the water we drink. So how much can you avoid it? And the problem is that we don't know. And until us as a community or our government starts coming up with ways to avoid these toxins, one of the things that does work in the short term is to overcome these obstacles with testosterone, right? And sometimes we have to push testosterone through these obstacles, right? So another thing that can prevent testosterone binding to the androgen receptor is the environmental toxins can cause an inflammatory response on the cell wall, right? So the cell membrane gets thicker and it inflames, which makes it harder for testosterone to diffuse through the cell wall to be able to bind to the androgen receptor, right? So again, one of the things we can do in the short term that does work is to push testosterone through these obstacles, right? And that may mean that some people need slightly higher circulating levels because we all have different susceptibilities or genetic predispositions. We all live in different environments and we all have different susceptibilities to those environmental toxins, right? So it's not about, well, this worked for me and therefore that should work for you. It's about we don't know what's going on with each individual. So it's about, sometimes we have to push testosterone through these obstacles, right? So in the conclusion, what does this all mean? Again, there are many complexities that we cannot measure. And because we cannot measure all these complexities, the numbers don't mean much, right? So again, when we're researching a protocol for ourselves, or when we're giving somebody else advice on a protocol, one of an important aspect to conceptualize is that we have to be able to see past our own self-centricity or an individual's response to testosterone, right? It's again, everybody's different, and everybody's different. There's a massive disparity between the way each individual reacts to different hormones, different blood levels, and different compounds. It's not about putting everybody in a box and saying everybody should be in a certain range or everybody should be on a certain dosage or on a certain protocol, right? Because of all these factors I went over, and again, 1,000 more that we don't even understand, 1,000 more factors that we don't even understand. There's no way we could put anybody in that box, right? So, so what it is about, and what I do at my company, hormonesforme.com, is it's about developing a methodology to figure out what yields the best results for each individual, despite all these variabilities.
Right. So, practical implication, what are the typical testosterone doses you see in your clinic, for example? What is the minimum you give to men, and what are maximums that you have been obligated to give?
Yeah. So that's a great question, and this will shock a lot of people, especially in the forums. there seems to have been like this, low dose evangelist that because the low dose worked for them, they go out and evangelize and say, no, this is what worked for me. You don't need that high of a dose. And what will shock most people is the average person, more than half of the people do feel better on doses of 200 milligrams or more. Again, there's so many different variabilities, and I've had people who have to do 400 milligrams a week to feel normal. We do have people on that kind of dosage, and again, it just depends on the individual. So again, it's about developing that methodology to figure out what yields the best results for each individual, and that starts with, we'll start at a moderate dose, right, and then slowly titrate up until there's symptom resolution, until we find the dose that again yields the best results for you, that makes you feel the best, while keeping an eye on all your markers, your health markers, for safety and long-term longevity.
Mm-hmm. Are there certain genetic tests maybe that people can do to check out whether their androgen sensitivity lies?
That's a good question, and there's nothing feasible, right? You can get a breakdown of your androgen receptor genetics, but at this point it's incredibly expensive, and it's not really feasible, right? And it doesn't necessarily. So what there are is the scientific community, they really only define disease states, right? Even though they admit, yes, there's variability between everybody, but they only really research disease states, right? So this is when your androgen receptors are vastly affected, right? And there's like, you have such a problem with your androgen sensitivity that it's causing like developmental issues, or it's causing major problems. So, but what has happened is, in the early 2000s, there was something like 400 to 600 known androgen receptor mutations, and it goes up every year. And now we're above 1,500, right? So we're always finding, what we've come to believe is, what they used to talk about is that there's a specific wild type. They call it a wild type. And they say, this is normal. This is a normal androgen receptor. This is what everybody's androgen receptor looks like. It's a 900 amino acid chain, right? And they said, here's a wild type. Here's what the normal one looks like. And people who have mutations may affect them differently. Well, now we're starting to realize that everybody's different. And androgen receptors are no different. There's variability in all these androgen receptors, and there is no wild type. Like, everybody's just a little different. And there's these specific domains that are important to the structure of the androgen receptor. And if there's a mutation in one of those important domains, then it can mess the whole androgen receptor up. But if outside of those domains, some of those mutations can just cause small differences. Like I said, maybe just like one less binding point, or just small differences, right? Or binding points to the coactivators, things like that. So no, at this point, there is no way to look at, because we don't even know all of the mutations. There's no way to do a test to see if you just have a slightly low androgen sensitivity, because we don't. Again, we're discovering new mutations almost every day.
Mm-hmm. Do testosterone esters influence the individual variability?
I'm trying to think if it would make sense that that would happen. I mean, because the esters, it's just a sense of we're cleaving off these esters to get to the base testosterone molecule, which our body actually uses.
Yeah.
And what it is possible is, what we have started figuring out is that we have enzymes that cleave these esters, but they don't cleave them necessarily one at a time. And there's different of these esterases that cleave the esters, and they cleave certain numbers of the esters. So for a cypionate molecule, which is an 8 carbon chain, you might have an esterase that cleaves 3 of them, and then one that cleaves 2, right? So depending on your individual predisposition to have those cleaving enzymes, then that different esters may affect you slightly differently than another.
Exactly. Yeah. All right. Well, Ryan, I found this very interesting. It's very a fresh topic to hear.
Yeah. Thank you very much. Again, thanks for having me on. It's great to have a platform to be able to allow people to understand all these things that I've been researching for decades.
Yeah, thanks for sharing. So you have your own TRT clinic. So tell us about it. How can people reach you?
Oh, you can just go to the website hormonesforme.com. Right now, I can only treat people in New York, Florida, and Vermont, but I'm working diligently to quickly add licenses. But yeah, we take this, we take all of these accounts, all these factors into account when we're treating somebody and developing a protocol.
Yeah, awesome. Sure thing. I'll link to your website in the description of this video. Again,
Great.
thank you so much, Brian, and talk to you next time.
Okay, Dr. Devos, again, thank you, and it was great talking to you again.
Leave a like and comment under this video and check out my new ebook, Acne on TRT.